The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
We're going to break down what is the general structure of amino acids. Now, amino acids have, as you can see on the screen right there, a carboxyl group and an amino group that are both bonded to the same carbon atom. Okay, now amino acids only differ from each other in their side chains of what we call their R groups. So what I'm going to do is I'll direct your attention to the bottom left-hand corner of the screen, where we have what is our generic amino acid. And as you can see, this generic amino acid right here has that carboxyl group right there, okay? And it also has this amino group right there. Now our side chain, that R group right there, is what actually distinguishes amino acids from each other. That side chain can be a bunch of different other groups, a bunch of different other structures bonded to the amino acid, which actually affect its properties, and for instance its acidity, its basicity, whether it tends to be hydrophobic or hydrophilic, etc. So the R chain, or the side chain right there, is a pretty important part of the amino group, but as far as the general structure of an amino group is concerned, you have what we're looking at right now, which is this right here. That is the general structure of an amino acid. Now, an alpha amino acid, we're getting more specific now, is one in which the amino group is actually bonded to the alpha carbon. Now, if you remember your very basic organic chemistry, you should be able to remember what the alpha position is. So the alpha position is basically the carbon, or the position that is attached to the highest priority functional group. So in an amino acid, the highest priority functional group would be this carboxyl group right here. So the alpha position would be the position first removed from that functional group, which would be this carbon right here. So that is the alpha position, and this carbon right here is referred to as the alpha carbon. So what then is an alpha amino acid? Very basically, an alpha amino acid is one in which the amino group, I'm talking about this group right here, is attached to the alpha carbon right here. So it's very basic. So we have our alpha carbon in that first position. That's our alpha carbon right here. And then directly attached to it is an amino group, and that will be our alpha amino group. And that's basically all we refer to, that's basically all we mean when we are referring to alpha amino acids. All right, now, what are proteins? Proteins are basically polymers of amino acids. Okay, so basically what happens in this case is you will have amino acids linking or joining together to form a much longer chain, a much longer string of amino acids that are bonded together, that is referred to as a polypeptide. Or if you like, you can also refer to as a polyamide. So poly meaning many, and peptide in this case means that we have many peptide links, as is the case in a polypeptide chain, or polyamide meaning we have many amide links, as is also the case in a polyamide chain. Now, what you have to remember is that individual amino acids will actually be linked or joined together by peptide bonds in this polypeptide chain. Now, how does this polypeptide bond actually form? So we're going to briefly look into how this occurs, because it's pretty important. And for that, I am going to direct your attention once more to the generic amino acid we have on the screen right here. So we have two ends to this amino acid. So if you were trying to lengthen it, okay, or to start a chain by adding more amino acids, we'd need to form a peptide bond between this one and, say, another neighboring amino acid right here. Just going to quickly draw out another generic amino acid. So what basically happens in peptide bond formation is you will have what we call the C-terminus or the carboxyl terminus, which is this end of the molecule, okay? That's the C-terminus, the carboxyl terminus. You'll have the carboxyl terminus actually losing that hydroxyl group, and you will have the N-terminus, okay? Or the amino terminus of the other amino acid right there, losing a proton right there to form a new link, which results in the elimination of a water molecule. So the amino group right here will lose a proton, and the carboxyl group of the other amino acid will lose a hydroxyl group, resulting in the elimination of water, and a new bond is essentially going to be formed between the nitrogen and that carbon right there, and that is what we refer to as an amide link, a peptide link. Okay, so a peptide link is basically a bond that is formed when the carboxyl terminus right here of one amino acid and the amino terminus of another amino acid join or come together with the elimination of water to form a new bond, a peptide bond or a peptide link. So it's a very basic concept, and that's pretty much how amino acids are able to come together and polymerize to form long polypeptide chains. And as you can see to the right of the screen right there, we have an example of what is a polypeptide chain. So you can see this is the bond that I was talking about, that we are referring to as the peptide link or the amide link, that is formed between the nitrogen of one of the amino acids and the carbon that was part of the carboxyl group in the other amino acid. So right there, in those two red rectangles right there, that's what we refer to as an amide link. Those are amide links. You can also call it a peptide bond or a peptide link, whatever you want. And what we have on the screen right here is basically a segment of a polypeptide chain. So at either ends or on either end of this chain, where you don't have any additional or any more amino acids, obviously at the C-terminus, that's what you'll have. It's going to look like that. And at the N-terminus, that will be another hydrogen atom right there. And if you wanted to further elongate or lengthen that chain, you could just simply catalyze an additional amide link or additional peptide bond formation at either end of that molecule, because as you have seen, it happens when the amino group loses a proton or a hydrogen and the carboxyl group loses a hydroxyl group. So that's pretty much how a peptide bond comes about. Now, proteins have a very diverse range of properties, and those properties confer a large degree of versatility in the functions which they perform. And that's actually what I want to highlight in the next slide. As you can see, proteins have a host of different compounds, a host of different functions.